Good afternoon, everyone, and thank you for your interest in this webinar series. We will be starting in a few minutes. As we wait for the other participants, we would like to remind everyone that you may use the Q&A box found below in the menu of your Zoom to type in your questions or concerns during the webinar. Thank you, and let us proceed in a few minutes. And good afternoon. Welcome to the day 3 of the PRPP FSRA 101 webinar. The topic for today is the role of risk assessment in public health. Here's the link for the attendance and pre-assessment form. That is bit.ly/FSRA101-pre-D3. For questions regarding the presentation or for the resource speaker, please type it in on the Q&A tab here on Zoom. Please mention your name and affiliation. For similar questions, you may upvote the ones that have already been asked, and they may be answered live later on by our resource speaker. Questions will be addressed after the lecture. The post-assessment quiz and evaluation form will be posted during the last part of this webinar. The chat box is turned on for all participants prior to the start of the session and during the quiz. We appreciate your feedback for today's webinar. Please feel free to chat your thoughts or comments during those specified times. Thank you very much, and let us all enjoy this webinar. We would also like to inform everyone that you may still view the webinar today for the next 24 hours once it ends through our PRPP YouTube channel, that is bit.ly/PRPP-YT. Once again, a pleasant day to all of you. We are glad that you're sticking with us during the third day of our webinar series. For our day 3, this webinar is entitled Food Safety Risk Assessment 101 or FSRA 101, Basics of Ensuring Food Safety from Farm to Fork, Tripartite Subseries. This webinar aims to capacitate individuals with knowledge and awareness on the general aspects of food safety risk assessment and is the second leg of the capacity building activities of the Philippine Food Safety Risk Profiling Project, or the PRPP. This is entitled Pagkaing Pinasafe, a series of capacity building activities to support Philippine food safety efforts webinar series. My name again is Desiree Cainco, the PRPP, and I'm humbled to be serving as your facilitator and moderator for today's webinar. Before we start, we would like to recognize the presence of our project leader for the Philippine Food Safety Risk Profiling Project, Assistant Professor Abigail S. Rustia. And of course, we are grateful to welcome the presence and participation of the representatives from the different agencies who are here with us today. We have representatives from the Department of Science and Technology, from the DOST PSHERT, from the DOST FNRI, from the DOST ITDI, and from the DOST regional offices. Good afternoon po. We also have representatives from the University of the Philippine System, from the Department of Agriculture, the BAFS, NMIS, the BIFAR, and the FSRA. We also have guests from the Department of Health, from the FDA, from collaborating non-government organizations such as the FAO and the WHO, as well as different collaborating institutions and relevant stakeholders. We also have representatives and participants from various state universities, private universities, and individuals from the different sector of the industry, all across the Philippines. Thank you very much for joining us. So once again, we are glad that you're taking interest in this webinar. Thank you for sharing with us your valuable time despite your busy schedules. Just a quick review of this week's webinar program. As you can see on the screen, this webinar will proceed with a lecture. After some time, we shall be having a 10-minute break. And then once the lecture finishes, we are going to have the question and answer session. Before we proceed, please be reminded of the following as we go on with the webinar. We would like to request everyone to rename your Zoom names with the following format: your agency or your office _ your name for ease of identification. We would also like to inform everyone that the whole webinar will be recorded for documentation purposes. Please fill up the attendance and the pre-assessment form and the evaluation and post-assessment form through the Google link that will be announced on the chat box. For a smooth discussion of the topics, we have automatically turned off your audios while the presentation is ongoing. For any questions regarding the topic, You may type it in at the Q&A box up with a similar question or raise your hand during the Q&A session so that we may unmute you and you can go and ask the question yourself. Please do not forget to mention your name as we shall address this question later after the lecture. A certificate of participation will be sent by email to participants who have signed the attendance and the pre-assessment and evaluation, as well as the post-assessment forms for each day of the FSRA 101. And that's it for the reminders. I'm sure we all are excited to learn from today's resource speaker. So now, please allow me to introduce our resource speaker for this lecture on the role of risk assessment in public health. Our resource speaker for today is a veterinary epidemiologist and poultry specialist in the Canadian Integrated Program for Antimicrobial Resistance Surveillance, or the CIPARS, Foodborne Disease and Antimicrobial Resistance Surveillance Division, Center for Foodborne Environmental and Zoonotic Infectious Diseases, Public Health Agency of Canada. Her line of expertise includes farm surveillance program development, data analysis, antimicrobial use metrics development, systematic reviews and meta-analysis on the poultry research coordination. She's also been a consultant and antimicrobial resistance project coordinator in the Food and Agriculture Organization of the United Nations, Regional Office for Asia and the Pacific in Bangkok, Thailand from year 2018 to 2019. She's also been the lead veterinarian for disease prevention poultry on the Ontario Ministry of Agriculture, Food and Rural Affairs, Guelph, Ontario from 2008 to 2009. She has also worked in private industries such as the Maple Leaf Foods, Hanover, Ontario, as manager of veterinary services and quality assurance from 2005 and 2018, and as a poultry veterinarian in San Miguel Foods Incorporation, Philippines from 1994 to 1997. Our esteemed resource speaker took up Doctor of Veterinary Medicine in the University of the Philippines in 1994 and continued with graduate studies from 2004 to 2006, including graduate diploma in avian pathology and medicine in the University of Guelph, Ontario, Canada in 2004. Diploma American College of Poultry Veterinarians in 2005, and Master of Science from the University of Guelph, Ontario, Canada. She has also been involved in numerous publications related to chemical and microbiological risks in food, such as topics related to antimicrobial use and resistance of microorganisms, including Salmonella, Campylobacter, E. coli in different chickens and turkeys in Canada. She has also been a part of the different international collaborative committees or partnerships related to veterinary antimicrobial use and resistances, such as the following: Canadian Veterinary Medical Association Expert Advisory Group, Pan American Health Organization, Poultry Sustainability Value Chain Roundtable, Canadian Animal Health Surveillance Network, Joint Programming Initiative on Antimicrobial Resistance, Antimicrobial Use Working Group, Poultry Health Research Network, and Industry Broiler Health Working Group in Ontario, Canada. Without much further ado, please help me welcome our resource speaker, Dr. Agnes Agunos.
Hi guys, can you hear me okay?
Yes po, ma'am. Good afternoon.
Thank you so much, Desiree. That was a very nice introduction, and I am so honored to be part of your webinar today, and I hope that you will learn from this presentation. Don't be overwhelmed with the number of slides. It's just going to be quick. So if I talk too fast, please do stop me.
All right, ma'am.
So can I control my screen, or do I need to share my screen?
I believe our fellow colleagues are doing the screen sharing for you, Pop, but please feel free to take control of projecting
so Okay. So I can advance the screen?
Yes, Pop.
Okay.
Thank you.
Thank you. Hi, everyone. Sorry, let's move back to the first slide number 2. I would like to acknowledge the CPARS team, the Public Health Agency of Canada, where I obtained some of my slides that we will be using today. FAO Philippines, the AMR program, and the Bureau of Animal Industry, Dr. Tony Rose Baroga and Marty Kaniwan, and photos coming from Dr. Joseph Rubin, my colleague here in Canada. Okay, the presentation today will be divided into 3 sections. One is introduction to AMR, the difference between food safety hazards, residues, and resistance, a little bit of the epidemiology of AMR in zoonotic foodborne bacteria. So we won't be talking about the clinical bacteria today. So this will just be focused on zoonotic foodborne diseases. As the second section is about the guidelines for risk analysis of foodborne AMR. Then the codex text, CAC-GL 77, 2011, and a risk profiling example. So I will be describing our experiences in profiling the Ceftiofur-resistant Salmonella Heidelberg. And the last section will be, hi guys. Okay, shall I advance the slides, guys?
Apologies for the bit of an interruption. We are just trying to recalibrate the settings here on Zoom. Please give us a while.
Yep.
I'm sure all of you out there, guys, are so excited to hear this lecture from Dr. Agnes Agunas. All right, okay. I believe we're back.
Okay, it looks like it's working now. So I think we stop at the third topic. So we are gonna be looking at the data that are available here in your country. If you could probably apply the codex text and foodborne AMR. Sorry. Okay, so at the end of this webinar, attendees will be familiarized with antimicrobial resistance, AMR in short, and its epidemiology with emphasis on the dissemination of resistant bacteria from farm to fork. So we will be examining the food pathway. Attendees will learn about basic principles of risk analysis of foodborne AMR, and attendees will be introduced to current AMR landscape in the Philippines and how risk profiling could be applied to address this national and global public health threat. So we'll discuss AMR, and so let's learn a little bit about what AMR is and the mechanisms. Okay, let's review the 2 types of hazards associated with the use of antimicrobials in animals. So there's 2 types of hazards, and they're not the same. One is antimicrobial residue. So this is a type of chemical hazard. This is when an antimicrobial drug or a metabolite is present in the meat, the egg or milk of an animal. And the tests are ELISA and a method known as liquid chromatography and mass spectrophotometry. And you have all these tests back there in the Philippines, the Bureau of Animal Industry and Bureau of Fisheries, Aquatic Resources. Intervention are to observe the proper withdrawal period so that the drug companies would recommend the length or duration between the exposure and the time when that antimicrobial is already gone from the meat, the egg, or the milk of the animal. So that means that the farmer or producer need to implement that period so that the maximum residue limit is, the residue that are left in the meat is safe for consumption. So it has to be below the maximum residue limits when these meat products or egg or milk are consumed. Antimicrobial resistance, on the other hand, so this is now the microbial hazard. is when a resistant bacteria or the genes are isolated from the meat or milk of an animal. And we need to isolate the bacterial organism and conduct antimicrobial susceptibility testing to determine if that organism is resistant to the bacteria. It has a higher public health concern because we all know that bacteria multiplies and they can be disseminated beyond the withdrawal period. So if a drug has a withdrawal period of 5 days, 7 days, or 31 days, antimicrobial resistant bacteria could persist in the environment much longer than the withdrawal time. And the intervention is antimicrobial stewardship. There are many definitions about antimicrobial stewardship, and the definition that I like best is the one coming from the United States Department of Agriculture. It says that antimicrobial stewardship pertains to the actions veterinarians and producers or farmers take to preserve the effectiveness or efficacy and availability of antimicrobial drug through prudent oversight and responsible decision-making and safeguarding animal, public health, and environmental health. Let's move on to the next slide. So this photo shows the epidemiology of antimicrobial resistance. So we'll see the different compartments. So the pharmaceutical compounds are produced and we consume this. So the animals consume that as well as our backyard animals that are in close proximity to our commercial herds and flocks. And also goes to aquaculture and also in crops. So you can see here the antimicrobial residues depicted in the red dots are the residues that are coming from animals and crops as well. And then there are these yellow dots. So let's say that these are the antimicrobial resistant organisms. So these antimicrobial resistant organisms coming from animals, when they consume the antimicrobials and bacteria develops resistance, goes back to us. So because we consume those animals, as well as backyard animals or from aquatic animals and plants. So this shows that antimicrobial resistant organisms can just move from one compartment to the next. It leads to a self-perpetuating cycle of contamination and recontamination in the environment. This is the AMR confusogram. So again, it's a depiction of the epidemiology of antimicrobial resistance. So today we will just be focusing on the food pathway. As you can see here, the circles are the compartment where antimicrobials is being used. So we have here our food animals, and we also have aquaculture, and then we have the process in between. So they go to abattoir processing plants, and then the meat goes into retail. And then we prepare and handle those products, and then it goes into human. So humans are also exposed to antimicrobials, as you can see in this figure. So the presentation will be focused on the food pathway today. This is another flyer from the WHO. It is also showing the cycle of antimicrobial resistance along the food chain. So here, antimicrobials are given to our food-producing animals of poultry or chicken, a fish, and large animal like cattle or hogs. So when they're exposed to antimicrobials, resistant bacteria develops, and then these are disseminated in the environment. So our animals are exposed to those as well as the producer or the farmer, and then it will end up in the food chain, and we consume those resistant organisms. So this is the food pathway. Humans, so people also consume antimicrobials. Now, if the infecting organism is already resistant to those antimicrobial, it will be very difficult to treat that disease. That's why we have to limit the usage in animals as well as the way we use this when we are sick. take-home message: AMR is a One Health issue and needs to be addressed in a collaborative manner. So it's not just the producer, it's not just the human health, but it has to be addressed in a One Health context, and we need collaboration for us to be able to control the different the different uh, the different pathways as well as stages in the production chain, so that we can ensure that foods that enters the food chain is safe for human consumption. So food safety enhancement and reducing the need for antimicrobials are some of the ways to prevent AMR. Let's move on to the next section. We'll talk about AMR in general. So antimicrobial and antibiotics. So these two terminologies are sometimes used interchangeably. So antimicrobial and antibiotic kills or inhibit microbial growth, yet results in minimal or no damage to the host. An antimicrobial is a natural or semi-synthetic or synthetic in origin. An antibiotic is natural in origin. So you might be aware of the penicillin. So it's derived from a fungi or a yeast. So that is a natural it's a naturally sourced antibiotic. So here in this figure, this was taken from one of my missions in Indonesia. So a farmer who has a layer farm also sells different types of antibiotic over the counter. So, and then there's no veterinarians on site to be able to give advice to whoever is buying those antibiotics from him. I think this is also very popular in the Philippines. You see a lot of agricultural shops selling antibiotics. Now, if we obtain those antibiotics over the counter and we don't really know their efficacy and what are their purpose, then we might be introducing a wrong usage when we feed our chickens or our pigs with those antibiotics. So that is also one way of encouraging antimicrobial resistance developing in our system, in our food systems and our environment. Moving on to the next slide. So antimicrobials are used in animals for treating, for treatment purposes. So now that involve sick animals. We are now treating the sick animals with antimicrobials, and that actually should be what the antibiotics are used for. Prevention is when we introduce antibiotics to healthy animals, so they are not sick, and yet we are giving them antibiotics, because we are not serious. Maybe one day they will be exposed to a disease and they get sick, and we will have a lot of animals dying. So that's, some producers are giving antibiotics to healthy animals to prevent disease. The last purpose or reason for antimicrobial use is growth promotion. This is also given to healthy animals, but at a lower dose, mainly to improve growth rates or production performance. Let's move on to methods. So this photo shown here is microtiter plates. So the broth microdilution technique is one of the advanced methodology for detecting resistance to antimicrobials. So this plate has different dilutions of antimicrobials, so a panel of antimicrobials, and they grow bacterial culture, and after 24 hours, they observe an inhibition of the bacterial growth. And that is how they determine if that bacteria is susceptible or resistant to that particular antimicrobial. And I believe here in the Philippines, the NMIS is now using this technique to characterize antimicrobial resistance from healthy food animals. Moving on to the next, so this is the antimicrobials that should be included in the panel. In most cases, the antimicrobials belong to highest priority, critically important antimicrobials. For example, the third and fourth generation cephalosporins, the macrolides, polymyxins, and quinolones. What it means is that these antimicrobials are being used in human medicine, and they are highest priority, critically important antimicrobials. So these should not be used in animals. And so for us to be able to detect that resistance is emerging in bacteria isolated from food animals, we need to test, we need to include this panel of antimicrobials into our methodology. There are also applicable national or regional guidelines for AMR testing. And the FAO has developed a guideline for testing organisms coming from healthy food animals. The WHO also developed guidelines for testing of organisms coming from human or from people. Another method is known as the disk diffusion technique. This is a relatively older method, but still used in veterinary and human medicine. It is maybe we can call it a forgotten art, but it's still very useful for informed decision-making regarding therapy of clinically sick animals as well as in human medicine. So the process involved the use of antibiotic impregnated discs. So these discs are impregnated with different antimicrobials, and this is a plate with bacterial culture. And then after 24 hours, they measure the diameter or zone of inhibition to determine if they are susceptible, intermediate, or resistant. The figure on the right-hand side of your screen is a plate culture of methicillin-resistant Staphylococcus aureus. And the antimicrobials here are beta-lactam antimicrobials, oxazolin, cefoxitin, and amoxicillin clavulanic acid. And because they are resistant to the antibiotics, you won't show any inhibition in this particular case. And the last method, these are now emerging methodology, metagenomics, and they are now being applied to attribute cases of infection that are like antimicrobial resistant infection, particularly in foodborne zoonotic diseases, and in some cases in clinical AMR infections as well. So in this particular publication, they used metagenomics-based approach for attributing sources of antimicrobial resistance determinants, and also for investigating the potential reservoir of those resistomes. So the figure on the right-hand side of your screen is detection of different genes. So these are different antimicrobial genes, and they have characterized the source. Say, for example, the veal, and if it's related to the turkey, and then the turkey, if it's related to human, or pigs related, and its relationship or its similarity to the human isolates. So this is how they would attribute the potential source of these different genes or antimicrobial resistance genes. Conclusion coming out of this paper, they said that the human resistomes, so resistome is just the group of genetic elements that are found in that particular reservoir. They said that they were similar to pig and veal calves than to those of the broilers and turkeys. And they are attributing the human resistomes to different livestock reservoir using the random forest. So they were able to characterize it using metagenomics and allow them to identify pigs as a potential source of AMR in humans. So this is now more of the advanced methodology. And this is used in countries with advanced capacity for AMR detection and source attribution. So going back to this particular figure, using metagenomics, we can now trace the particular source of those resistant organisms infecting people. So if it's coming from animals, if it's coming from fish or aquaculture, from crops or the environment. So that is it has its utility for investigating antimicrobial resistance on a One Health concept. Take home message number 2. for investigating AMR in foodborne zoonotic bacteria isolated from various food animals, consider a panel of antimicrobials that are important for treating infections in humans. So for us to be able to attribute AMR happening in human population, there should be a similarity in the antimicrobials that are to be tested relative to those that are likely to be consumed by the animals as well as in human population. Moving on to the mechanisms. What really is antimicrobial resistance? So antimicrobial resistance occurs when a normally susceptible bacterium grow in the presence of antimicrobials at levels which normally suppress growth, or we call it bacteriostatic, or kill the bacterium, bactericidal. A minimum inhibitory concentration, or MIC, is the lowest concentration of an antimicrobial at which the bacteria is inhibited from growing. So as you can see in this figure, we have these circles. So the green circles represents growth of the organism. So these are growing in lower concentrations, and the white circles are zones where the bacteria is inhibited. So these are now the higher concentration of antimicrobials. Moving on to the next slide, there is also another thing that we need to consider here. It's the breakpoints. This is the dilution or concentration that if the bacteria grows above it, so if it grows above this, between 2 and 4, it means that the bacteria is resistant to antimicrobial in this example. And anything that grows below it is called susceptible. Now, if the bacteria grows between 0.5 and 1, then any isolates that grow in that dilution and above will be classified as resistant. So now they are becoming resistant to lower concentration. So in reality or in practice, if we have a particular bacteria that's being used a lot, or it has been used in an improper manner, like a penicillin, and we use a very low dose and very short period of time that the organism develop resistance, we might need a stronger concentration for that bacteria to be effective in treating that infection. In simpler terms, antimicrobial resistance can develop genes for resistance through mutation. They can also pick up genes for resistance from other bacteria, and the genes responsible for resistance may cluster together and move between bacteria as a unit. And AMR also developed through two different mechanisms. One is intrinsic or natural resistance. So intrinsic means that is a trait that is shared universally within a bacterial species. So it's their nature, it's their natural ability to resist ability to resist antibiotics. It is independent of previous antibiotic exposure, so regardless of the exposure, they are showing resistance to that particular antibiotic. And it's not also related to horizontal gene transfer, so that is their natural trait. Acquired resistance, as the name implies, is acquisition of genetic materials that confers resistance. And let's examine this in more detail. One is intrinsic and natural resistance, meaning that they lack the cellular mechanisms necessary for susceptibility. In this particular example, gram-positive organisms and polymyxin B. The mechanism of action of polymyxin B is to disrupt the phospholipids in the cell wall. However, the gram-positive organism doesn't have the phospholipids. That's why the gram-positive organisms will never be susceptible to polymyxin B. So these are examples of the gram stain of Staphylococcus aureus and a plate of Staphylococcus aureus. So Enterobacteriaceae here on the right-hand side of your screen, and vancomycin. So the vancomycin disrupts the phospholipids in the cell wall. So they are supposed to be effective only for gram-positives. So if these are being used to treat a gram-negative organism because they don't have the cell wall, and this is only present in small population of gram-negative organism, vancomycin will never be effective in treating gram-negative infection because gram-negative bacterial organisms are intrinsically resistant to this particular drug. Also, vancomycin's molecule is so large that it won't be able to cross the cell membrane of any gram-negative bacterial organism. So that should not be used for treating gram-negative clinical infection in both animals and humans. Again, acquired resistance. There are two types. One is chromosomal mutation and transferable resistance. For chromosomal mutation, there are two types, stepwise and one step. Stepwise means there are a series of events leading to resistance. So genetic changes in the chromosome of the organism. So this happens in penicillin and resistance to fluoroquinolone. And one step, so you need only one single mutation for that resistance to happen. For example, for streptomycin, one single mutation could lead to a 1,000-fold increase in minimum inhibitory concentration. Transferable resistance, that means that there's a transfer of genetic elements happening between bacterial organisms. So HGT, or horizontal gene transfer, happens through different mechanisms like transformation, transposition, and conjugation. And they might also experience mutation to its own chromosomal DNA. Plasmid-mediated, as shown here, is the most, shown in the photo here, is the most common route for acquisition of outside genetic material. In case of, if you are interested, these are some of the transferable resistance going on. So bacterial transformation, meaning that it only happens in certain bacteria. So there is a release of naked DNA or antimicrobial resistance genes, and there should be a good homology of the recipient cell to the donor cell. And then there's an uptake of those antimicrobial resistance gene in the host cell happening. So transduction is just the release of phage. So you need a bacteriophage to be able to transfer that DNA genetic element. And this is also very rare. Bacterial conjugation, so that is when a plasmid is moved between a donor cell and a recipient cell. So there is a pilus created, and a plasmid is transferred, a copy of the plasmid is transferred to the recipient cell. And the plasmid might contain multiple genetic determinants. It can be up to about 7 that is moved from between the different cells. There are more detailed explanation of this different transferable resistance that you can acquire via the web as well. So this is how to explain the horizontal transfer of genetic elements. For example, in the host gastrointestinal tract, when we are infected with a non-pathogenic bacterium that's carrying a resistance gene, and we're also infected simultaneously with an antimicrobial susceptible pathogenic bacterium. So this is the bad bacteria or the bug that is really bad, that's causing the disease. The other one is non-pathogenic, however, it's resistance. It might be happen when we're consuming, say, a contaminated chicken, a contaminated pork. So these different microflora might be present in the meat. Now, what happens is when we are infected with the non-pathogenic bacteria that has the resistance gene, they will multiply inside the guts, because of course that's the nature of the organism. They multiply inside the host cell. Now, if we introduce this bad bacteria with which has the virulence genes or very pathogenic, there is now a transfer of the AMR gene to that particular organism. Then they multiply and it would emerge into an antimicrobial resistant pathogenic bacterium. So not only that it is pathogenic, now it is also becoming resistant. So now if we are sick with this infection, say for example, Salmonella organism that is resistant to beta-lactam antimicrobial or multiple classes of antimicrobial, we try to treat that infection with the same antimicrobial, beta-lactam, as cephalosporin antimicrobial, for example, then it might take a while before we get the desired effect. And the efficacy of that drug will now be ineffective or very low because the infecting organism is already resistant and we are not responding to that infection. Take-home message number 3. The ongoing and improper and widespread use of antimicrobials in people and animals speed up the emergence of antimicrobial resistance.
All right. I believe we are mid-lecture right now. Perhaps it's a good time to take a short break. Would you agree, Dr. Agnes?
Okay, yeah, we can have a short break before we continue on the main theme, the AMR risk assessment.
Yes, let's give a 5-minute break to our participants, and we will be open to hear your initial feedback here in our chat box. Please feel free if you're experiencing any technical difficulties here on Zoom, you may let us know and we shall see you. It's now 2:48 on the clock, so let's meet at 2:53. All right. Thank you and see you all in a bit.
Once upon a time, the Earth was a huge playground for us bacteria. Those were joyous days. We were wild and free, helping or hurting people and animals as we wished. Then one day, humans invented antibiotics, a weapon that took this power from us and put them in control. We were no longer free to cause pain, disease and death among humans and animals. But we waited. And humans made a fatal mistake. They used these precious medicines indiscriminately in humans, plants, and animals. And some of us realized we could develop a defense to these antibiotics. Humans call this phenomenon antimicrobial resistance. We call it taking back control. Now we, the disease-causing bacteria, can spread wider and faster where disorder, dirt, and disorganization reign. It's great for us when you have poor hygiene and don't use water and soap. We love when you flush your unused antibiotics down the sink or toilet. They join our already huge colonies in your wastewater and river systems and make us stronger. Poorly managed hospitals are good for us. In these places, we're free to multiply and become new ruthless killing machines. Antibiotics given to animals not to control or treat disease, but to make them grow faster, create new armies of bacteria that become increasingly resistant. The beautiful thing is that the simple contact between infected animals and humans can allow us to spread. We can also circulate in contaminated food, on plants, in the soil, and in water. We can be everywhere. We've regained some of our old powers. However, I am becoming very worried. Our enemies are starting to work together. International organizations and countries are starting to coordinate all sectors, from human, animal, and plant health to agriculture and food safety, to halt our glorious expansion and stay on top. They explain to people that we can be stopped by using antibiotics, as per doctor and veterinarian's advice, and not for growth promotion, by adequate wastewater management, and by having proper hygiene and cleanliness at the hospital and at the farm. Humans could become wiser and keep us down. A disaster. Imagine if everyone listened to their advice. I am no longer safe. My ability to resist antibiotics could disappear. Don't let antimicrobial resistance take control. Be responsible when using antibiotics.
That was that was indeed an interesting infomercial about antimicrobial resistance. And thank you for Dr. Agnes Agunas for sharing that with us. Before we proceed again with the lecture, may we just remind everyone um, that for questions regarding the presentation or for the research speaker, please type it in on the Q&A tab, and please don't forget to mention your name and affiliation. We shall be answering those questions once the lecture is um, is done. So, all right. I hope we are all now ready to resume the lecture. Please have the floor again, Dr. Agnes.
Okay, I need to advance my screen to the section where we'll.
All right. Uh, let us please have the current slide. We stopped at slide number 28.
Okay, yeah, we can start here.
Thank you very much.
Hi, everyone. So this is the second section of the webinar. So the role of risk assessment in public health and the guidelines for risk analysis of foodborne AMR. So there are two key documents, key reference documents for AMR, for foodborne AMR risk assessment. So one is the CACGL 77 adopted in 2011 here, the Codex documents, and the Codex text and foodborne AMRs. Basically, these are complementary documents. Let's move on. So this figure is coming out of those two documents, the framework for foodborne AMR risk analysis. And you might be familiar with the process already. Some of these were discussed yesterday. And we will be focusing on the identification of an AMR safety issue and development of a risk profile for AMR, because this would align with the mandate of your team of the project. Also, I want to draw your attention here The surveillance of AMR, so surveillance of antimicrobial resistance and other sources of information is critical. So it's integral to all the different components of the risk analysis. First is to identify the AMR's food safety issue. So there are preliminary activities that needs to be done to be able to identify or prioritize what that AMR food safety issue is. And there are multiple sources of information according to the Codex text. This can be coming from outbreak investigations of foodborne illnesses, monitoring and surveillance. So this is coming if you have a national antimicrobial resistance and antimicrobial use programs. Those are the sources of information. Research, existing AMR risk assessments, scientific expert opinion, and stakeholder and consumer feedback. For example, there is a recall of particular product contaminated with resistant organism that's causing a lot of foodborne illness, and it's causing a huge or significant burden of illness in the country. What are the purposes of a risk profile? So one is to provide the current state of knowledge regarding the food safety issue and identified risk management options. So this provides the state of science by conducting a scoping review of all available information about the topic or the particular food safety risk. Then to put the issue into context, so contextualizing local conditions and production setup. Last is to make recommendations for the following types of decisions. One is an immediate or provisional decision for risk management. So you might need to implement something that is drastic. Then do you need to launch a full risk assessment? And are you going to be doing a qualitative or a quantitative risk assessment? Do you need to gather more data to conduct a risk profiling exercise or just maintain a status quo? So according to the Codex text, these are the elements for consideration in a foodborne AMR risk profile. Number one is description of the AMR food safety issue. Two is the AMR microorganism. So we need to identify the bacteria and the particular determinants of resistance. Number three is the information and the antimicrobial agent to which resistance is expressed. So what is the antibiotics or antimicrobial implicated? Number four is information on the food commodity. Is this a chicken? Is this a pork? Is this a beef? Is this an egg, a milk, or a fish? Information on the adverse public health effects. Is it causing high mortality or high morbidity in human population? And what is the demographics? What's the age implicated? The risk management of information. How do we manage that particular risk? And seven is the evaluation of available information and major knowledge gaps. Take-home message number 4. The purpose of a risk profile is to provide the current state of knowledge regarding the food safety issue and identified risk management options, or RMO. So now I'll take you back here in Canada, and let's examine a risk profiling example that's conducted here using the Codex guidelines. So we will be describing this particular publication. So this article was published in Epidemiology and Infection, Septifure-resistant Salmonella enterica, serovar Heidelberg of poultry origin, a risk profile using the Codex framework. Basically, my role here is to describe the commodity in question. So I'm a poultry veterinarian and I'm very familiar with the Canadian poultry industry. So that component is my piece of contribution to this particular manuscript. Number 1, description of the AMR food safety issue. Sorry, let's go back to the last slide. So So it's the Ceftiofur-resistant Salmonella Heidelberg. So we identified this based on the defined combination of three components. The resistance hazard, so what is the resistant microorganism or determinant? So that is the Ceftiofur-resistant Salmonella. The antimicrobial agent is of course Ceftiofur, a third-generation cephalosporin, and the food commodity is chicken. So this is a graphical example of the AMR food safety issue. So Salmonella Heidelberg is a serovar that tends to cause more illness, severe illness in people compared to other Salmonella serovars. So you might be aware that there's about 2,000 Salmonella serovars, and most of them are foodborne. In Canada, we also have a diverse serovar of Salmonella. But this particular serovar is causing more severe illness compared to other Salmonella serovars like Enteritidis, Newport, Kentucky, Reading, Hadar, or other or Typhimurium or other Salmonella serovars that are common in North America. And the antimicrobial to which resistance is expressed is the third-generation cephalosporin. So you will hear me talking about Ceftiofur or ceftriaxone. So they all belong to the third-generation cephalosporin class of antimicrobials. And as I mentioned in the previous slides, that this is a high priority, critically important antimicrobial for the treatment of various infections in people and animals. Chicken is the meat source commonly consumed by Canadians, and it is the source of Salmonella Heidelberg. Moving on to the information again on the AMR microorganism or determinants. So there is a figure on the left-hand side of your screen. So this figure on the y-axis, the vertical, is the percentage. So this is just the proportion, or sometimes we call it prevalence. And below are the different commodities. We have the retail chicken, the retail breaded chicken, or chicken nuggets, turkey retail, abattoir chicken. So this is the processing plant chicken, farm level, and farm turkey. So we have a data coming from the different sectors of the poultry industry. And there's four different information here. Briefly, they are just the recovery or percentage of samples that tested positive to Salmonella. Among the positive Salmonella isolates, how many of those were resistant to Ceftiofur? The teal colored bar, and then the green is the percentage of Salmonella Heidelberg. So among the Salmonella serovars detected, how many of those were Heidelberg? And among the Heidelberg, how many of those were resistant to Ceftiofur? So this just shows that most of the resistant isolates are coming from chicken. So just in case, we also took samples from swine, so the pigs, from retail, the abattoir, and at the farm. However, we did not see any resistant Salmonella Heidelberg, although there's a lot of Salmonella coming out of those swine samples. So in this case, we are ruling out swine as the potential source of Salmonella Heidelberg. The figure, the pie chart on the right-hand side of your screen is the distribution of Salmonella serovars from chickens. So as you can see here, the most frequently isolated serovar is Kentucky, followed by Enteritidis and Heidelberg. So Heidelberg is just the third most frequently isolated serovar, but this tends to be causing a lot of illness in people. Number two, again, this is a temporal trend. So again, in this figure, you would see the prevalence of Heidelberg here on the vertical axis. This is the year of surveillance. So in other presentation that I will be giving 2 weeks from now at the TVMA, I call this the Great Canadian Experiment, because the industry is conducting their own large-scale experiments. So, there's multiple things happening here. The blue series is the retail chicken prevalence of Salmonella Heidelberg resistant to the antimicrobial. We have turkeys as well, later part of our surveillance, beginning in 2013. And we also have human isolates here, depicted by the red series. So because we have an integrated surveillance system that we can now collect information from multiple components, from animals as well as humans. And we also have some antimicrobial use happening, data coming from the farm. And as you can see here, there's a strong temporal correlation between the resistance that we saw in retail chicken isolates as well as in humans. So it's highly conclusive that the infection in humans is originating from chickens. And it also follows the different important events happening in the industry. So the data coming from 2003 and 2004 initiated the public health action. And they have to withdraw the use of ceftiofur in poultry. So we have seen that the drop in resistance. So it shows that when you take something out, you don't use it, you see a drop in resistance. No, they decide that they would want to use it again. So they reinstituted the use of ceftiofur. And so we see that emerging resistance, it emerged again. And then suddenly it's becoming so prevalent that they need to implement another public health action. So that resulted to the elimination of that particular antimicrobial. So in short, we use this data to conduct our risk profiling exercise. Now, what do we know about the antimicrobial? So cephalosporins, as I mentioned, are beta-lactam antimicrobials. They are bactericidal and they inhibit the bacterial cell wall. There are 5 generations of cephalosporins, so 1 to 5, so based on their spectrum of activity, this particular antimicrobial is 3rd generation cephalosporins. And then they have increased activity against gram-negative bacteria, such as the Enterobacteriaceae, but decreased activity against gram-positive bacteria. So going back to our lesson about intrinsic resistance. Okay, this slide is showing the non-human uses of the drug. So in Canada, in some parts of the world, this drug is injected in hatching eggs. So these are the hatching eggs that are incubated to become the chicks. Now in human, this is used for a variety of bacterial infections involving the respiratory tracts like pneumonia, urinary tract infection, skin and soft tissues, and intra-abdominal organs and the central nervous system. That's why this should not be used in this animal species in chickens. As you can see here, it's being used in human medicine. So we try to preserve the efficacy of that antimicrobial so that when we are sick with this particular diseases, that this will remain effective. Information on the food commodity. Just like here in the Philippines, chicken is the most frequently consumed meat in Canada. So each Canadian is consuming about 35 kilos of chicken per year. So this picture here on the right-hand side of your screen is a farm in the Fraser Valley in British Columbia. It is a very nice place to grow chickens as well as grapes. Now on the adverse public health effects of Salmonella Heidelberg. So in between 2003 and 2011, there were about 638 cases of invasive Heidelberg infections in Canada, and 30% of those occurred in children under the age of 13 years old. And I want to emphasize here that the Heidelberg, even if they are foodborne, they're also very invasive, meaning that these were detected from the blood, so they can cause septicemic disease. And so in 2016, Heidelberg accounted for 7% of all reported human salmonellosis cases in Canada, despite that there are many serovars infecting people. So it's very significant in terms of the burden of illness associated with foodborne diseases in Canada. And these are the percentages as well. It could vary depending on the time frame. And I want to emphasize that it always coincides with the use of the 3rd generation cephalosporins in chickens. So we can see that association between the use in chickens and the diseases in humans, . Sorry, my slides are advancing on its own. Okay, number 6 is risk management options. This was taken from the same article, but you can maybe revisit the article for better resolution. So it just shows that there are potential intervention, and there are challenges as well for implementing this different intervention. And as you can see here, there are relevant stakeholders in each aspect or stages of the poultry production continuum that are involved in implementing this intervention to reduce the prevalence of that particular serovar, as well as the prevalence of the resistant salmonella. Sorry, guys. So these are the risk management options. So let's talk about the parent stocks. Parent stocks is just the parents that produce the hatching eggs. So unlike the eggs that we buy at the grocery store, so those are coming from layers, they are non-fertilized eggs. So here are the parent stocks. It's just like maybe in your grandmother's yard, you have a cockerel as well as the hen. So they are laying eggs that are fertilized. So in this case, in commercial farms, we see a lot of the hens here. Like it's a massive, large scale commercial farms. We have hens, and then there's a nesting system where they lay their eggs. And these are the eggs that are sent to the hatchery. So there's a process. Here in Canada, we get those hens when they are like baby chicks. We grow them to be pullets for 20 weeks, and they lay for up to 40 weeks. So here is the laying barn where they lay the eggs. And then they are just sent at the abattoir when they are done laying. So there are multiple steps that are implemented here by the industry. They are monitoring the use of antimicrobials. They're vaccinating against salmonella. They have on-farm food safety compliance protocols. And they are also required to implement biosecurity and pest control to ensure that the eggs are free from any salmonella organisms when they are hatched. And this is the farm, the poultry production chain, so from broiler breeder to consumer. Then the hatcheries, the broiler producers, we have the processors, the distributors, and retailers. So these are the different stages in the poultry production continuum. So here in Canada, we have the boards. So these are groups of producers that manage the volume, as well as the food safety programs in the different sectors. So they implement the on-farm food safety program based on HACCP. And there's a lot of measurements to ensure that the food or the product produced are safe for human consumption. And as this transition from one stage of the production continuum to the next. So there's these food safety audits, there's monitoring of antimicrobial use and resistance. And in some cases, like at the broiler level and hatcheries and processors, they are conducting routine monitoring or microbial testing as well. The QMRA. So QMRA is quantitative microbial risk analysis. So a lot of you are probably working on some projects involving QMRA. So this particular example is an offshoot of the risk profile. So we look at the different stages of production from parent stock, the hatchery, up to the consumer, and to evaluate the risk to people. So this particular exercise can answer different questions, like what is the risk? What is the estimate of the risk? And if I want to implement a change or an intervention, how much can I reduce that particular risk? At what stage? So in this particular exercise, we know that these are the most critical intervention by stopping the administration of antibiotics in chicks at the hatchery level, that one. And we need to change antimicrobial use as well, behavior, to reduce the dose response, to impact a dose response. And another measure measures are preventing the prevalence or reducing the prevalence of third-generation cephalosporins resistant salmonella from entering the food chain and preventing salmonella in general. And there are some other interventions as well pertaining to the production of chicken. So those are also very important in reducing the risk, preserving, and the cases or the burden of illness in people. Then let's examine this again in more detail. So this is the paper. Okay, so there's this one particular figure coming out of the QMRA. So you all see this particular gray bar. So what does it mean? So these are the scenarios. So each of these number are pertains or explains a scenario in the model. So this in your y-axis, the vertical axis is the average probability of illness per random serving of the intervention scenarios. So these are the interventions from 1 to 19 to the baseline scenario. So let's just say this, the probability of illness. The baseline is status quo. You don't do anything. Number 2 to 10 are farm level interventions. Number 11 to 14 are processing intervention. 15 is retail. 16 to 18 is home, and 19 is the hazard. Now this is the human. So these are the different phases from farm to fork. And then you can see that there are some areas where we see very low probability of infection, but there are high ones. So let's move on to the next slide and what those numbers really mean. Okay. Probability of illness on your vertical axis, and these are the different scenarios. This one, if we reduce the use of antibiotics at the hatchery, we are reducing the probability of illness by 90%. The worst case scenario, we don't do anything, we'll keep on medicating the chicks or exposing the poultry with antibiotics. So this would also happen in a lot of cases, not only in this particular scenario, like ceftiofur. It can be an amoxicillin, a penicillin, a tetracycline being used in pigs or in chickens. So this can just be replicated in multiple ways, but it shows that if you don't do anything, this is the worst that could happen. You will increase the infection in humans. Now, these are all maybe important, but it could fluctuate depending on what the intervention is at the farm and at the processing plants. And here is also very significant, as you can see here, reducing temperature abuse at retail, meaning that when you store the chickens, the storage temperature needed to be monitored. In the Philippines, this is probably an issue that needs to be also to be improved, because I know that there are wet markets. So how do you manage the temperature there? And under tropical conditions and exposed environments, we are probably multiplying the growths of microorganisms if they are starting with high level of contamination. And then number 17 is now pertaining to our behavior at the household level. So we prevent undercooking and proper washing. It would reduce the probability of illness by 65%. And there's also a responsibility of the community or in people. We need to be cautious about the way we use antibiotics, because if you already have exposure before you are infected, then there's also you are increasing we are increasing our probability of illness. Whereas if we are not exposed to that particular antibiotic, it would reduce the probability of infection by about 65%. So this is just a simulation using the data that we have so far, but it shows that if we implement steps in relevant phases of the production chain, we can really reduce the infection or burden of illness in people. And then you have seen this from Dean's presentation yesterday, the dose-response relationship. So a basic explanation is here. So the gray bar is you have previous exposure to the same antibiotics before infection. And then this one, you don't have previous exposure. And this is the log colony forming units. What it means is that you have been exposed. You only need very few organisms for you to become infected or become ill. Whereas if you don't have particular exposure, then even if you have a higher ingested amounts of bacteria, your probability of illness is much lower. Take home number 5. A risk profile generates sufficient evidence to inform risk management options or to enhance our existing interventions across the food production continuum. And the Codex framework provides a useful approach to summarize data for decision makers to evaluate an AMR food safety issue. At this point, would you like guys to continue on or take a 5-minute break to digest those particular slides?
Yes, ma'am. We believe we could use a 5-minute break once again.
Okay.
Thank you.
So
To all our
let's go back then.
Yes, to all of our participants, please feel free to use this time to give us a few of your thoughts, share with us your experiences so far, and let us see you all again in a bit. In the meantime, we are delighted to acknowledge the presence of the Dean of the UP College of Home Economics, Dean Shirley Guevara. Thank you very much, po, for gracing us with your presence this afternoon. We would also like to acknowledge our esteemed webinar resource speakers, Mr. Dion Mahani, Dr. Hans Marvin, and Dr. Yamin Buzenbrock. We're also glad to let everyone know that the DOSTP shared representatives, Ms. Fatima Ibarreta, Ms. Chelsea Ugay, as well as their colleagues are here with us today. We also have our dear guests from different collaborating bureaus under the Department of Agriculture, Department of Health, the Food and Drug Administration, the DOST regional offices, and all our colleagues from the University of the Philippines system across the country. Once again, see you in a bit. Thank you. And now for the final stretch of our lectures, let's once again have Dr. Agnes Agunos. I hope everyone is ready. All
Okay,
right.
we'll start. Okay, thank you.
Thank you.
Thank you guys for staying. This is the last section of the presentation. So now we talk about the potential application of risk profiling for foodborne AMR in the Philippines. So what I did was a mini scoping review and gather information as much as I can from the internet. So what is the state of science, agriculture, and AMR in the country? Moving on. So the state of science to evaluate risk for the Philippines. Are the existing frameworks enough? But will this be applicable under Philippine conditions? Do we need to contextualize and very specific and develop a flexible approach under our conditions? Because I know that there are resources that might limit our ability to evaluate the risk for AMR. Do we have data or the right data to use? When we have the data, and what do we do with it? Number two, series of questions. Why do we want to evaluate risk of AMR from Philippine agriculture? Do we want to know where problems are, or how do we compare it? So is it across sectors, commodities, or between countries? Do we want to know whether something is a risk and by how much? Do we need to do something, and what interventions will be the best, and where do you need more data? There are different data that are available and different ways of putting the data together. So we've seen this, but we won't be talking about this anymore. So we will be examining the information that you have available in the country using the same elements that we discussed in the previous section. Number one, description of the AMR food safety issue in the Philippines. So I put it here DBC, so the drug, the bug, and the commodity. In terms of foodborne pathogens, are we interested in characterizing foodborne salmonellosis, salmonella infections, extended spectrum beta-lactamases, or Campylobacter? What is the antibiotics or antibiotics to which resistance is expressed? What is the commodity implicated? Is it a chicken, a pork, or a beef, or eggs, or other products? Number two, information on the AMR microorganism. Number one, so I have here a traffic lights. So I color-coded them into yellow as if yes, maybe we can proceed. There's a little bit of information. Crops, no, we don't have any data. And green is yes, we have a lot of data. So starting with animals and food of animal origin, including aquaculture. There is no global reporting yet of antimicrobial resistance in food animals. There are several national and regional surveillance programs. So here in the country, it is on its very early stage. So the NMIS is developing their capacity and working with the WHO and FAO in developing their methodologies. And often we are characterizing Salmonella, E. coli as indicator organism, Campylobacter with or without Enterococcus. So Enterococcus is a gram-positive AMR indicator used in many surveillance systems across the world. Animal pathogen. This is a very big gap, so we don't see a lot of AMR data coming out of pathogens from animals. With regards to crops, there's no data, there's very limited information. And the scoping review conducted by the FAO found that there's 25% of products contaminated with bacteria with more than one antimicrobial type. And in humans, the GLASS is a global reporting system, and there's 92 countries in the latest reports that is contributing to the GLASS AMR data. And it includes Salmonella and E. coli. So it doesn't have the full range of pathogens. Information on the AMR microorganism and or determinants. Now, animal and food of animal origin in the Philippines. There are few AMR research projects in food animals in the Philippines. So later on in the next two slides, we will be looking at the highlights of the project conducted in Central Luzon, in chicken, as well as in pigs. AMR surveillance program, early stages of the AMR surveillance program is on its early stages of implementation, and I know the NMIS, and I think some of them are here today, are developing their capacity for surveillance of AMR from healthy animals. Animal pathogens, there's also a pilot project, but it has very limited information as well. And we hope to gather a lot of data coming out of the animal pathogens because they are an indicator if the treatment at the farm level is not working, or the range of antimicrobials that are available for treating these diseases may not be working to treat those particular pathogens. Okay, this is an example of a research project that was done in the Philippines. The table on the left-hand side of your screen is the distribution of ESBL genotype among ESBL producing E. coli. So ESBL are extended spectrum beta-lactamases. These are enzymes that would degrade penicillin as well as third generation cephalosporins. So they would develop this capacity to produce this enzyme when they are exposed to these antibiotics for a long time. So this is just showing that there are diverse ESBL genotypes contaminating the chicken coming out of those farms that they survey. And among the different genotypes, there's the CTXM. The CTXM is the most prevalent type of beta-lactamases that are circulating in humans as well as in animals. So the concern here is that if these genetic elements would transfer between animals to humans via the food chain, then there is a possibility that it might cause an AMR-associated illness in people. So these are some plates of extended spectrum beta-lactamases here grown in selective media. And this is electron photomicrograph of ESBLs. Another example, so the same authors also examined the ESBLs from pigs. So these are from swine farms. And I want to draw your attention on the left-hand side of the screen, and I'll see if I can zoom this particular section. Okay, so as you can see here, these are multi-drug resistance patterns, meaning that if I take one isolate, what are the resistance that I would be likely to find? So for example, in this particular isolate, there's 9 isolates that exhibited resistance to penicillin, cephalosporins, fluoroquinolone, and I think this is fluorphenicol. And so there's about 4 different classes of antimicrobials to which this organism is resistant to. So in most cases, like these guys, there's even five classes. And in the same isolate, it's showing resistance to cephalosporin and fluoroquinolone. So if you could remember that these are very high priority, critically important to human medicine, and this should not be used for treating diseases in animals. Because as I mentioned, if these plasmids or these determinants would move from food animal to humans, then it will be very difficult to treat this infection. Okay, and then on the right-hand side of your screen is also showing the diversity of CTXM. So they also have these different CTXMs in pigs, similar to chicken. And I also want to draw your attention here. MCR1, this is the colistin resistance gene. So it confers resistance to colistin. And again, colistin is very highly high priority, critically important antimicrobials, and it is used in a variety of diseases in people. So we should be cautious in using these particular antibiotics in animal production. But here in this particular research, 26 of the isolate out of 48 had this particular genetic determinant. Next, we move on to humans. AMR microorganism coming from human isolates. So I was able to extract this data summary report published by the Department of Health, the Antimicrobial Resistance Surveillance Program Summary, 2019. And this is the percent resistance of non-typhoidal salmonella. And these are the different antimicrobials. This is the prevalence of resistance from 0 to 40%. These are the different antibiotics. So just a little bit of background again. So we have your ampicillin here, the cefotaxime, ceftriaxone are all beta-lactam antimicrobials. The cotrimoprazole is a combination of trimethoprim and a sulfonamide antimicrobial. It's also a very popular drug for treating infection in humans. The levofloxacin and ciprofloxacin are fluoroquinolone, and you are aware of the chloramphenicol as well. And these are the prevalence. So high prevalence to ampicillin, so about 30%. And you also have some resistance to the fluoroquinolone, less than 10, and the beta-lactam antimicrobials. Moving on to the next slide, also from humans. The pie chart on the right-hand side of your screen is the Salmonella serovar distribution. I also extracted this information coming from this report. A vast majority of the isolates were typhi, 51% of 198. And half of the non-typhoidal isolates, or half of those are non-typhoidal isolates. So among the NTS or non-typhoidal isolates, Salmonella enteritidis was the most frequently isolated serovar in 2019. So we know that Salmonella enteritidis is always foodborne, at least here in North America and parts of Europe. They are usually associated with poultry, so poultry meats and eggs. So it is likely that these are like foodborne illnesses coming from isolates. These isolates are coming from foodborne cases submitted to the Department of Health. I think it's the RITM who does these isolates. Now, I was also able to find this information. You might be aware of this. So in January, the Department of Health announced their capacity for genomic surveillance. So this is a huge milestone for the Philippines because genomic surveillance is very rare in low and middle income countries. So this is going to be used for source attribution. So it says in the news release, they have moved into a genomic era, and it will enable a better tracking of dangerous bacteria. So meaning that they can now attribute the source of those resistant organisms to their true sources or potential or likely sources of infection. Another milestone. So the Department of Health also published this in Nature. So Nature is a very high impact journal. So any scientist would like to publish here in this journal. So the application of whole genome sequencing. So their capacity for whole genome sequencing is highlighted in this particular manuscript. And an example is shown on the right-hand side of your screen. So using the isolates coming from their own archive. And I think this is ST410, so Salmonella, or it's an E. coli. And here they were able to create a phylogenetic tree, comparing the different genotypes, resistance determinants to their sources, to geographical locations, so they can link epidemiological information like geographic origin of the isolates and what are the resistance patterns. And here in the lower half, they could also compare with various isolates collected from all over the world. So sometimes if there are cases that are travel associated or might be related to an imported product, then now they have the ability to detect those the source of those resistant organism. It's really cool because you have this capacity now and you can do source attribution. Moving on to the antimicrobial agent to which resistance is expressed. So I went and examined the PVET. So this is a compendium or a database of all the products used in food animals in the Philippines. And according to this database, there's so much third-generation cephalosporins, the fluoroquinolones and quinolones, colistin, and combination drugs that are used in these animals, in food animals. So meaning that we need stronger regulation to manage or maybe restrict the use of these antimicrobials because these are used in humans. So there should be a more restricted access to this particular antimicrobial class because we want to preserve their efficacy. So the Philippines contribute data to the OIE, sorry. But the OIE reports is still not stratified by country. So the antimicrobial use is reported at the regional level. The national reporting of antimicrobial consumption is stratified by antimicrobial class and species is still a big gap, but I know that the Bureau of Animal Industry is now working towards the reporting of the antimicrobials that are used in animals. So in the future, you will be able to know the proportion of antimicrobials or the antimicrobial class being used in a particular species, as well as the relative quantity of the antimicrobials, and be able to document it over time. Now, there's one pilot project conducted in the Philippines, in chickens as well as in pigs, and the study population was both commercial and backyard. And these were the information collected coming out of the survey. From chicken, these are the 5 most frequently consumed or frequently used antimicrobials. There's a lot of fluoroquinolone use, and there's also colistin use. In pigs, it's the same. There's enrofloxacin, and there were also some cephalosporins reported. So meaning that we still have to work towards the reduction of these antimicrobial classes in food animals in the country. In humans, we don't have a Philippine-specific report set, but the Philippines contribute data to the WHO, the reports and surveillance of antimicrobial consumption. So these reports. So the national reporting of antimicrobial consumption stratified by antimicrobial class is also a big gap at the moment. So we don't know yet the extent of antimicrobials consumed in human population, as well as the classes that are being used. On the commodity, so the meat products that are consumed in the Philippines, I extracted it from these statistics reports for 2020. Pigs, it shows here that pigs is the most frequently raised or consumed animal species, followed by chicken. You also have a lot of layers, cattle, and then the different ruminants, have the goats, carabao, and dairy. There's also some ducks and duck layer in the country. So it shows that these are the very important commodity in terms of maybe potential reservoir of antimicrobial resistance. Now moving on to, just showing the temporal trends in production. So this is in metric tons. We see a steady increase in the production of pork. I don't know with the COVID if this trend had decreased or not, or remained stable. So pork and chicken production are increasing, I think, over time in the country. And the information on the adverse public health effects in humans, that is not routinely reported. We don't really know the impact of the public health implications of illnesses that are associated with resistant organisms. However, there are many indicators of burden of illness, but it is very difficult to summarize the information, and we don't know if those are truly related to AMR. Say the length of stay, intensive care unit admission, treatment failure, mortality, and hospital costs. There are also different comparator groups. Do we want to know the different demographics or different geographical locations? Dose response gap, and what about prior human AMU? So that sometimes is difficult to determine. Even if we have a detailed case information, we may not know the exposures to antimicrobials in humans that are infected with AMR. Animals, we probably have to request the industry to provide us with those information like production indices, like feed conversion rate or growth rate, and those other types of measurements pertaining to growth and production of animals, and maybe mortality rate as well. So with regards to risk management information, it has to be specific to the animal or the species, the sector or production stage. And there's multiple references also. It can be based from other countries, just like our exercise here in Canada that have similar production structures. So we can always replicate those production practices that might have a sparing effect or could offset the need for antimicrobials. Evaluation of available information and major knowledge gap. So what I want to say here is there is a window of opportunity because there is a global call to contain antimicrobial resistance. There is a Philippine action plan to combat AMR. So this is a plan, it's a sectoral plan, and it identifies the areas where they can reduce the impact of antimicrobial resistance, or they can contain the dissemination and spread of AMR. So it might be an opportunity to gather more information as well as to implement some interventions to reduce AMR in the country. Because you have some data that are available that you can leverage to be used maybe for a risk profiling exercise, risk management, or just basically of reducing the antimicrobial in poultry production so that you can preserve the efficacy of the antimicrobials that are available for the treatment of both animal and human diseases. For tools, there are lots of models that are available out there. They can be qualitative. So a risk profiling example could start as a qualitative exercise, moving on to semi-quantitative or quantitative. So the information that I presented a while ago is an example of a quantitative modeling, QMRA, and there's also an integrated assessment model. And that is only possible if you have an integrated surveillance system and multiple sources of information that you can use. And of course, the approach will also be dictated by availability of the data, the resources available to model. So if you have the right team and the right knowledge, expertise to conduct the exercise or the modeling, and the time available to conduct it. Then the last two slides are just questions that you might need to consider if we want to explore conducting a risk profiling for AMR. We know that there is a framework, but we need expertise for using new types of data. Can we use whole genome sequencing if that is now being used here in the Philippines? Do we have standards for other routes of exposure? So not only through the foodborne, and how to make recommendations from the data that we have gathered. There are some relevant data, but there are also many gaps. So here in the Philippines, for example, the salmonella data that I was able to extract, we need more isolates to better characterize AMR in certain serovars that are likely going to show resistance to the antimicrobials that are being used in humans as well as in animals. Other foodborne pathogens like Campylobacter. So I think the human data is still lacking Campylobacter AMR. AMR in crops in animals also is needed. AMU in crops, animals, and humans. And human data, we need to have dose response data that might be very difficult to obtain. The burden of illness as well. So how much is the government spending in reducing the foodborne associated illness in the Philippines? And other questions, what do we do with the data and how do we develop the models? We also need recommendations. There maybe other countries could help the Philippines direct the approach taken to be able to address this question, and how to link the different sectors. So there should be a very strong linkage between the researchers, the industry. There's multiple sectors of the industry, as well as the governments and researchers that are conducting surveillance, and how to move into prediction as well. So what do we do with the simulations and how to contextualize those results? And what do we do with results and where this is all going? Do we need to change behavior? And how could we translate it into standards? Or how do we enhance the guidelines that we have here in the country? And could it be used to leverage fundings for a sustainable integrated antimicrobial use and antimicrobial resistance surveillance program in the country? So is there a need to enhance the current capacity we have so far? Finally, last slide, conclusions. Food animal commodities are frequently contaminated with foodborne bacteria resistant to antimicrobials of importance to human medicine. So those are coming out of the research studies that are published or available. And there were many out there, but I did not have the chance to extract all the information, but to have some more information that are available, highlighting that some of the foods in the Philippines are contaminated with AMR organism, particularly from the meats or food animal industry. In humans, Salmonella isolates were also resistant to the same classes of antimicrobials used in animals, so the third generation cephalosporins and fluoroquinolones. And it is only a matter of time to link the animal and the human AMR pathway, because now you have genomic surveillance to be able to attribute the source of AMR. A framework exists for AMR in foodborne pathogens, so the Codex guideline is available as a reference. And there are also some experiences from other countries. There are also some data available in the Philippines for potential risk profiling. This could then inform enhancement of food safety and public health measures, and possibly to seek political commitment to implement components of the AMR National Action Plan. I think that's the end. Thank you so much for your attention, and I will be, I think I will have to address some of your questions at that after the break.
Thank you very much, Paul, for that, Dr. Agunos. It was a very informative lecture, and it's been truly enriching learning experience for all of us to be a part of this webinar with you. Please know that we shall be having another 15-minute break, after which we shall have the question and answer session. So thank you, Paul, and we'll see you in a bit. To all our dear participants, and for the benefit of those who might have missed it, let us once again share the following reminders regarding the Q&A session. So throughout the webinar, the attendees can ask questions through the Q&A tab only. Please make sure that whenever you type, it's on the Q&A tab. Please refrain from asking through the chat box. Also, raise similar questions will be upvoted so that there will be more chances to have been answered live by the resource speaker. A moderator will facilitate the Q&A portion for the webinar, and a participant may raise their hand to be unmuted and to be allowed to ask the question themselves. Otherwise, the moderator will be the one to read the question. Unanswered question by the resource speaker due to limited time left for the Q&A may be answered by the panelists via the Q&A tab. Kindly take note of these reminders for a smooth discussion in the Q&A portion later. So, all right, let's now have that 15-minute break. The time now is exactly 4 p.m. We shall resume by 4:15 p.m. to open the floor for questions from the participants. You may also start typing your questions right now on the Q&A tab of this Zoom platform, which can then be answered later after the break. Thank you once again and see you all in a bit. Welcome back. How are you all doing? We hope that you are all able to freshen up during the break, and we are all very excited to hear the questions you have in store for our research speaker this afternoon. But before we proceed with the question and answer session, let us first have a quick wrap of the things that we have learned this afternoon. For today's webinar, we have been provided with a background on antimicrobial resistance, or the AMR, its epidemiology, development, and distribution in the farm-to-fork chain. In relation to food safety, we have also learned about the guidelines for risk analysis of foodborne AMR, specifically on the elements for consideration in a foodborne AMR risk profile. Sample application of risk profiling foodborne AMR to address a food safety issue was also discussed. And lastly, profiling of foodborne AMR in the Philippines was discussed, wherein we have learned about the current state of science, available local data, and existing framework for foodborne AMR. It's been very amazing, Dr. Agunas, how you were able to explain such complex technical information into very comprehensive bits that are very easier to understand. So we're truly grateful for. All right, I believe we are now ready to open the floor for questions. So, there will be an allotted time of 15 minutes for the Q&A for an estimate of 5 to 10 questions to be accommodated. For the rest of our participants, please feel free to type in your questions on the Q&A tab. Alternatively, you can also opt to use the raise your hand function here on Zoom so that we may unmute you and you can ask the question yourself. While waiting for other questions, we can start by having some of the questions that are already answered. Please allow me to read them all. There is a question from the DOST 12 from Samira Makabalang. The question is, can these two references, the Codex Alimentarius, be used as standards for test method on antimicrobial assay for accreditation by the Philippine Accreditation Bureau? To which our research speaker answered that these are only applicable to antimicrobial resistance from bacterial organisms. There's also a question from BAFS LSD. As I understand, local surveillance data, records of adverse effects in our country, and local studies on dose response are needed for developing risk profiles. If these are not available, then can we develop risk profile? Please confirm. To which our research speaker answered, risk profiling can still be applied upstream of the food production continuum. It answers questions like what is the estimate of the risk, but more importantly, what are the options to decrease the risk. It is still informative in directing specific interventions. The dose response exercise comes later. So thank you for those questions. We also have another one from She Bilango, Balaro. In the northern part, the question goes, in the northern part of the Philippines, one of the traditional dishes makes use of chicken. In the early times, backyard-grown chicken were used, but it is no longer a practice because of the availability of broilers or cull from the lowlands as substitute. Usually, the chickens have reached their maximum laying period and is brought to the different stores in the highlands because of the high demand. May I know your thoughts of our overconsumption of this chicken could possibly be exposing us to the risks of AMR? Thank you very much. And with that, our research speaker has answered, acknowledged the question, and said that they are aware, or she's aware of the spent costs, and that overconsumption is not the issue. It is only when these chicken products are contaminated with resistant organisms and we are handling them and cooking them improperly, such as in the case of undercooking, improper washing, and incorrect storage temperature. There is also another one from Michael Daniel Ramirez. The question goes, are readily available over-the-counter drugs contributing to the rise of AMR? As people tend to self-medicate when having a cough, fever, or body ache without knowing the correct dose and frequency of intake, how do we address this? To which our research speaker has answered, yes, that the over-the-counter drugs are an issue in both humans and animals. There is a need for modernizing the regulations to restrict access of these antimicrobials, OTC. I am aware that pharmaceutical industries are aggressively promoting their products and the pharmacies also want to sell them. The healthcare system also needs to be improved to support access to medical doctors and affordable antimicrobials, but this will be very challenging if it is not a priority by the country. So far we have those answered questions. May we please entertain now another one from a user. The question goes, um, Dr. Agunas, hi. What about risk assessment studies for shellfish and other marine animals?
Thank you for the question. I think it is possible. So you need also to use the same process. You need to identify the AMR food safety hazard or the issue. So what type of organism is implicated? What is the specific commodity or what type of shellfish, right? So we need to characterize those, and are those associated with antimicrobial use in aquaculture. So those information we needed to obtain, but it is possible.
All right, thank you po for that answer. And to the user who sent the question, thank you for that question. We also have another one from Farlash Pancho. Hi, Dr. Agunos. May we know how if there are specific guidelines on the prioritization of commodity being subjected to a risk profile? And for someone who is new to it, can you provide us a background on the specific relevant details that need to be reported in a risk profile report? Thank you.
Thanks for the question. For prioritizing specific commodity, there really isn't. So I would just consider what is the most frequently consumed commodity or meat commodity in the country, and then where did you identify the hazard? So it is a hazard. It is a combination of the hazard, so the drug, the bug or the organism, and the commodity combination. So you need to assess those. Those are the three different information that you need to know before you can implement a risk profiling exercise. And then the guidelines. So we just follow the codex text. So the 7 items that I listed in both presentations or both sections, you can use the same patterns in conducting the risk profile. So there is already a framework that exists for AMR risk profiling, and just getting the details or information we need to populate the different components, I think that will provide guidance on how you could conduct the risk profiling exercise.
Thank you po for that. That's been a very informative answer. We also have another interesting question from one of our peers here in the PRPP team. Dr. Agunos, question from Mr. Edjul Paul Lindero via our YouTube live stream. Is resistance to antifungal drugs a cause for concern?
Thank you. So the antifungal drug also, it would target, of course, the fungal organisms. So we have infections that are also that also need antifungal drug therapy, that they can be systemic or localized, like the toenail fungal disease, and there are some ear infections, and there are systemic infections as well, like aspergillosis is a respiratory disease. And then those would need antifungal drugs. So then that is a separate discussion as well. So the mechanism for fungal resistance is a little bit different. So the antimicrobials that are useful for fungal therapy may not impact the resistance that's happening in the foodborne diseases or bacterial organisms. So there's 2 separate mechanisms.
All right, thank you very much po for that. And now as we wait for a few more questions, since we have a bit of a time to spare, can we at least hear from our participants of their thoughts and some of their other expectations for the rest of the webinar series? You can also share some of your insights. It doesn't have to be a question, but you can also spark a discussion with us, we can use this opportune moment to discuss with our resource speaker this afternoon. As everyone would know, all of our resource speakers are esteemed experts in the field of food science from across the globe. So it's really a big opportunity for us to be a part of this webinar series. We have another one from our YouTube live chat from Irene Bethalas. The topic is very interesting and it encourages us to be involved in research to better provide safer foods for the Filipinos and perhaps improve our food safety practices. It's more of a comment. Thank you very much po for that, Miss Irene. It's been a truly enriching afternoon. And for our second day of our webinar, it's really exciting to see the same attendees. We're getting repeated attendance from our participants. So on behalf of the whole of the PRPP and our project leader, Assistant Professor Riz Chow, we are truly grateful to everyone who's been a part of the webinar series so far. And we hope that we can continue it for the last stretch of the webinar series for tomorrow with another of our experts in the field. Can we hear more questions or comments from our other participants here on Zoom and on YouTube? We still have a bit of time to spare, but if there are no more questions, and well, if our research speaker would like to have any last few thoughts.
Thank you, everyone. Thank you so much for the time and for your attention. AMR is a very complex issue, and some of the information that I shared today, these are just bits and pieces. It's a full expertise, a whole discipline to learn about AMR. It's epidemiology, it's biology, surveillance is another topic. So a lot of things that are that needs to be learned. So if you still have questions, then maybe just channel it through the PRPP team, and I would be able to share more detailed response.
Yes, thank you very much for that. And I believe we can have our one last question for this afternoon from Segundina Devoto. The question goes, can meat inspectors and sanitary inspectors be trained as risk managers?
It is possible, but I'm not really sure, depending on the extent or what is their involvement in managing the risk. So they probably can provide a lot of feedback as to the processing steps and areas where they can improve. So they can provide the practical practical information where risk managers could use in developing the risk options for managing that particular risk. In terms of implementation, I think they could also play a big role because they are the ones that are interacting directly with maybe the owner of a processing plant, the producers who are supplying that particular abattoir or processing facility. There's some kind of dynamics that needed to happen. So if they provide a feedback to individual producers, and then provide feedback to the owner of the processing plant, provide feedback to research team. So there are multiple ways that could maybe, I don't know, that they could assist in managing, providing risk management options.
Indeed.
Dr. Agnes, can I add something to it?
Yes, please.
Okay, thank you. So for that questions, I think for the risk managers in the country relative to the food safety, risk managers are the Department of Agriculture, which has a regulatory function, and from the FDA, the Department of Health, who has a regulatory function. So mostly risk managers comes from that area. And I think, as what Dr. Agunus said a while ago, they can provide essential information that will help us clarify information and the exposure assessment pathway. Yeah, thank you.
Thank you very
Thanks,
much
Abigail.
for that. And I believe this wraps up our question and answer. Thank you very much, Dr. Agunos and Assistant Professor Juscha, and to all who have taken part in this certainly enriching discussion. We thank you for joining us this afternoon. And now, to continue on, let us once again have the project leader of the Philippine Food Safety Risk Profiling Project, also known as the PRPP, our very own Assistant Professor Abigail Ruscha, to present the Certificate of Appreciation to our resource speaker this afternoon, to Dr. Agnes Agunas.
Cellphone. So, good afternoon, everyone. I would like to present the Certificate of Appreciation to Dr. Agnes Agunos in appreciation of her invaluable contribution to the Philippine Food Safety Risk Profiling Project webinar entitled The Role of Risk Assessment in Public Health, as a source speaker and public health expert. This seminar is a part of the tripartite webinar series, Food Safety Risk Assessment 101, Basics on Ensuring Food Safety from Farm to Fork, of the Capacity Building Initiative Pagkaing Pinasafe, a series of capacity building activities to support Philippine food safety efforts webinar series, held this 4th of February 2021 via online platform. Signed by yours truly, A.S. Rustia. Thank you very much, Dr. Agunos, for your help and then for all the information that you shared with us. Thank you.
Thank you very much din po, ma'am, for presenting the certificate. And once again, thank you very much, Dr. Agunas, for sharing with us your knowledge and expertise about the role of risk assessment in public health. Indeed, it has been a privilege to learn from among the most brilliant experts in the field, such as you po. We would also like to thank our participants for giving their inputs for today's webinar. And of course, our participants, for you to receive a copy of your certificate of participation in today's webinar, please do not forget to answer the post-assessment quiz and evaluation forms. The link is already up in the chat box. And please make sure to accomplish the form for your e-certificate that will be sent directly through your registered emails. Your responses in the evaluation form are very much appreciated and will be used for further improvement of future PRPP webinars and workshops. Before we go, may we request the members of the PRPP team, together with our project leader, Assistant Professor Ruscha, the DOST Assured representatives, Ms. Fatima Ibarreta and Ms. Chelsea Ugay, and the UP College of Home Economics Dean, Dean Shirley Guevara, and the rest of the PRPP team, and of course, our resource speaker. Please turn on your videos for a photo opportunity. Once again, we are inviting the members of the PRPP together with our guests. May we please turn on our videos to have a photo opportunity with our resource speaker in today's webinar. Let us just wait while everyone gets ready.
Let's go ahead.
All right.
Ready? One, two, three.
Another one, please. 1, 2, 3. All right. Thank you very much.
Hey guys.
There we
Thank
go.
you.
Yes po. Thank you very much. Thank you to all of your invaluable support to the PRPP. Lastly, to know more about the PRPP and to be updated with our latest activities like this webinar series, you may also like the PRPP Facebook page. We also have the PRPP on Twitter. It's at Risk Profiling PH. You may also feel free to subscribe to our PRPP YouTube channel. And for any questions, concerns, and suggestions, you may reach us via the social media accounts and via our PRPP Gmail. We hope that you can join us again tomorrow for the last day of our webinar series. The topic for tomorrow is about big data in food science, big data in food safety, and food fraud, to be delivered by our esteemed resource speakers, Dr. Hans Marvin and Dr. Yamin Guzenbrock from the Wageningen Food Safety Research. Once again, thank you, stay safe everyone, and see you all again tomorrow.
Thank you, Des. Thank
Thank
you,
you very
everyone.
much.
Yeah, thank you.
Thank you po. We shall be ending the meeting for all in a short while. Good afternoon, sir, from Bell Computer Center. May we please request that the meeting be ended for everyone po? Thank you very much. Good afternoon once again to Sir Will of the Computer Center. May we please request that the meeting be ended for all? Thank you very much po.
Hello? Tapos na ba kayo?
Yes, sir. Thank you po. Nalipat po sa inyo yung host ko. Ay, ayan. Okay, thank you everyone.